Top Med Talk.
Hi, and welcome to Top Med Talk. We're here in EBPOM, London 2023. I'm Desiree Chappell, your host, and I'm joined by my co-host, Monty Mythen. Hello, Monty.
Hey, Desiree. Good to be here. Day 3 of EBPOM London 2023 back at University College London, and I think it's been a great success.
Yeah, I think so too. It's been a lot of fun. I actually haven't gotten to enjoy all of it. I've been in session for POCI 11, Perioperative Calling Initiative number 11. If you are interested in more of that, please do check it out on the podcast. There's lots about it.
An update on fluids, blood pressure levels, and goal-directed therapy, whatever that means.
I think we're going to learn more about it here in just a minute. So it was a very interesting 2 days, intense talking about, specifically in my group, goal-directed hemodynamic management. And chair of that group is here to join us today, Mark Edwards. Hello, Mark. How are you?
Hello. I'm good. Thank you. I'm good. I'm kind of tired, but also excited. And it's been a great few days.
Yeah. So POCI was great. And like I said, we're actually going to have another conversation about that. Why I'm actually having you sit down with us today is something that you've been really excited about. And I was shocked at how composed you were over the last couple days. There was a big announcement that you made here at EBPOM this year. Again, tell us, Mark, why you're here at EBPOM today.
Today was the kind of early reveal for the results of the OPTIMISE II trial that I've been working on with the chief investigator, Rupert Pearce, for the last probably 10 years, from conception and initial designs and funding attempts through to recruitment and finishing recruitment last year and analysis. And we were very happy to share the results with the EBPOM conference and also live stream that to the international investigators around the world. So that was the first time the results have been released publicly. So that was good.
Well, we're going to dive into that in just a little, in just a bit. Mark, tell us, for all of our listeners, tell us a little bit more about yourself and then, kind of how you got involved in this trial.
Thanks. I'm an anaesthetist and researcher based in Southampton, UK, on the south coast. I first got involved with OPTIMISE II, invited to help lead it by Rupert back. And as I say, it was about 10 years ago now. OPTIMISE, the first OPTIMISE trial, and this is a topic area that Rupert's been working on for many years. OPTIMISE was finished, was reporting. It was the biggest, until now, the biggest trial of perioperative, goal-directed, cardiac output-guided hemodynamic management approaches that had been there. And as I'm sure many listeners are aware, it was a sort of minimally invasive cardiac output-driven intervention in elective surgery, mostly elective surgery, and had got really close to showing some benefit, like P equals 0.07.
Rupert's folly, as he refers to, and we've talked to him about that.
In the interest of time, we did a very detailed interview with Rupert at the European Society recently, where he told that story brilliantly. Yeah,
it was really, really good. So we can definitely refer back to that.
The thought was that there was a signal. They were probably a bit underpowered. Perhaps they just needed to refine the intervention a little, refine the patient group, go again with a bigger sample size, and just really try and do that definitive trial.
Yeah. So OPTIMISE II was born out of that. So started 10 years ago. Tell us a little bit. I think it's okay to maybe dive right into the...
Just want to touch on a tiny little bit of detail about OPTIMISE I, because it came out in Mark's presentation earlier today.
Yeah.
As often happens in all of these trials when we attempt them, we very rarely take exactly what we did before and scale it up.
Yeah.
So because of the passage of time, correct me if I'm wrong, OPTIMISE I was still relatively restrictive maintenance fluid, as I remember. It's a similar group of patients, very similar group of patients. Stroke volume optimization was a core principle, and there are a few different ways of doing that. It was done predominantly, I think, with a colloid at the time because of the era that we
OPTIMISE
were in.
I, colloids were the recommended fluid bolus.
And the main drug to stimulate the heart was dopexamine, which is pharmacologically subtly different in a way that may or may not be relevant to what was used in the subsequent one. So the subsequent one has some subtle differences. And you just touch on them for a second, because they may or may not be relevant. It will be one of the discussion points.
So you talked about dopex. Obviously, we're scaling up, pragmatic, international, needs to fly in a variety of healthcare systems. We decided to give a choice We're aware that Dopex might not always be available, might not always be within clinical utilization in those places. So we said, okay, it's either dopexamine or an equipotent dose of dobutamine. And just to pick up one point you made about it increasing cardiac output, that's an interesting question, because this was a pretty fixed, pretty fixed low dose that was used. And there is a lot of thought, and a lot of Rupert's team's prior work actually suggests that there's maybe an important effect on gut microvascular function, tissue oxygenation, possibly even immunomodulation. So whether it's a kind of macro hemodynamic effect, or whether actually at that lower dose, the idea is with these other, perhaps slightly more subtle effects, is that's out there for discussion.
Dopexamine, I remember, it sounds like we're already making up excuses here, was more dopaminergic in its characteristics than the more commonly available dobutamine, which is more beta adrenergic.
Yeah.
And pharmacologically, that's quite different, and it may or may not be physiologically quite different. But it's the element, the nature of pragmatism, as dopexamine was not commonly available. I don't think it's even is available now, is it?
I've not seen it in the UK for some time.
Okay.
It's not in the US.
Yeah.
Colloid versus crystalloid difference, and the drug different.
Yep. Other minor points: slightly shorter post-operative duration, 4 hours instead of 6. Again, can this fly in the real world?
That was for the dopamine, or this not dopamine, sorry, dobutamine infusion was.
That was the whole package.
Yeah.
So we still wanted people to be getting assessments of fluid responsiveness and stroke volume targeting.
I gotcha.
In the early post-operative recovery,
gotcha.
Along with the infusion.
Going. Okay.
And then the last thing, I guess, was introducing stroke volume variation into the algorithm, but just as a break.
Okay.
So Neil McDonald's nice work in those grey area values. We're not quite sure that it adds a lot to predicting fluid responsiveness. But at the extremes, so a super low value, less than 5%, we can be pretty confident that next fluid challenge would not increase your stroke volume. So don't give it. So it actually allows you to have a break and not necessarily go on with that next unnecessary fluid bolus.
And the optimization of the stroke volume ran through the duration of the case. That was an ongoing. That was the ongoing physiological target, was the optimization of stroke volume.
Yeah,
and therefore a suggestion that optimizes cardiac output, even though cardiac output, which is a combination of stroke volume and heart rate, was not a number target. So you, as far as I understand, you didn't say give me an index of 2.5 or a DOU of 600.
Correct. Yeah, it was see what you can get the stroke volume to until there are no more signs of fluid responsiveness, and then stick.
And run the dobutamine from the get-go unless you get too tachycardic.
After the first bolus. Got to get a bolus, at least one bolus in the system. And then, the dobutamine starts. And there was a protocolized ability to turn down the infusion or stop it if they got a tachycardia.
Yeah, I mean, my next question was going to be, let's walk through the algorithm a little bit, because I think that's important to do, especially, is there a fluid bolus up front? Not a fluid bolus, but I mean, is there just a fluid load at the beginning of the case? Then you start doing fluid challenges, or is it, how does all that work? So I think let's just start at the beginning and walk through it a little bit.
Right. So an encouragement to start monitoring as soon as possible after the initiation of GA, to start with the 250 bolus given over less than 5 minutes.
I'm sorry, I'm going to stop you. A-line always, or was there?
Not necessarily.
Okay.
So we were using Edwards devices, and either the FlowTrack invasive, if an art-line was being used, or the finger pulse ClearSight could be used as an alternative.
Gotcha.
Interesting question. I need to go back and see how many were using the ClearSight. I haven't actually seen that, so it's a good reminder. Thank you.
There you go. No problem.
They would have been majority art-line.
Yeah.
For sure.
Okay.
Yeah.
Major elective surgery is what we're talking about here.
So
abdominal surgery.
First bolus, and then we're going around these cycles to see, has there been a 10% or greater increase in the stroke volume? If yes, we're going around the circle, but you only go back to the top of the circle if your SVV remains above 5%.
Mm-hmm.
Okay. But anyway, let's say it does. You think you're still fluid responsive. You go around that cycle until you get a value of stroke volume, which has not responded, or your SVV has gone below 5%. Mm-hmm. At that stage, you say, okay, let's stick and watch. And if that is your stroke volume number that persists for the next 20 minutes, the idea is you can be reasonably sure you have optimized that stroke volume. You've achieved what the maximum it can be at this stage in proceedings. Now, we're having some interesting discussions in POCI. Hang on, does that really mean you've maximized cardiac output? Yeah. Not really. If you wake that patient up, send them to a 100-meter sprint, they're going to get a higher cardiac output. But at this stage in proceedings, from where they are in their process, that's the kind of maximum value you can achieve.
And that was it. There was no cardiac output targets. There were no blood pressure targets per se, just to avoid it.
So yes, we had a blood pressure target, although we did not protocolize how you got there.
Gotcha.
So
Okay.
we had for all patients, both the intervention group and the control group, we had broad hemodynamic targets. So
Okay.
we said, please maintain a map between 60 and 100 using whatever your usual care would dictate. And we saw about 40% of participants in both groups receiving infusions of vasopressors/inotropes, presumably to hit that target. But again, we weren't didactically...
Out with the protocol.
Correct, yeah.
So I could choose to start a phenylephrine or epinephrine infusion to keep the blood pressure up.
Yep.
And I wouldn't need to turn it down to determine whether it's having an impact on my stroke volume or...
Correct.
We recognise these are challenges in all the studies.
Yeah, absolutely.
These are not criticisms. These are just
No.
recognising
I'm just trying to,
how they do or
right.
don't match on to how we practice. And about 40-50% got an
infusion.
Another vasoactive infusion.
Yeah.
In
Additional.
both groups.
Correct. Yeah, out with the trial.
So again, sorry, there's no
initial time. Yeah, go.
When you presented the results today, Mark, we don't give too much away apart from the headline that's out there. Randomization works.
Randomization was good. Yeah.
So
randomization with a minimization.
The number again of the final groups.
So N that were included in the intention to treat analysis and had a primary outcome available was 1247 in each group.
Great.
Yeah. So we were gunning for 2,500. We're pretty much there. Really good primary outcome availability, which was great.
And the big surgeries we'd expect them to be. We don't have the time really, but the mix of ASA2 being relatively common, some 3s and 4s in there.
Yeah.
Duration about 4 hours.
Yeah.
4 hours of
4 hours.
surgery.
Yeah.
Blah blah blah. They look extremely well matched.
Yeah, absolutely.
But my next observation would be the therapeutic interventions that both groups received seem to be extremely well matched as well.
Yeah, interesting. I mean, very similar fluid volumes.
I know there is a difference in one got some dobutamine and one didn't, but for everything else they got, they seem to get treated the same way.
Yeah, and I mean, I do just observe that over the years, the fluid volume differences in goal-directed hemodynamic trials, the fluid volume differences seem to have shrunk down. And I think in the first OPTIMISE, it was in the order of 250 to 500 mils difference. There are other trials where, again, 500 is often touted as, this tends to be the difference, and the different paradigm is around, has the timing been different? Because there must be some individualization somewhere. But yeah, so it was a small, 100 and some mils difference intraoperatively, and that's about a sort of 2.5 liters to 3 liters overall amount that both groups got. And then again, in the 4 hours after surgery, about 600 to 700 mils.
The difference between the two, as you said, is about 100 mils.
Yeah, 150 mils off the top of my head, something like that.
So something unlikely to be physiologically relevant. We could have the timing discussion.
The timing thing, yeah. And
which will all come out in subsequent analysis.
Yeah, of course. And same fluid types.
Yeah.
So overwhelmingly buffered crystalloid. Okay.
Lactated ringers, Hartmann's type of thing.
Yeah, plasmolyte. Yeah.
And the bolusing, I mean, not like necessarily when it happened, but the amount of fluid boluses that each group got were similar or different?
It would have been, yes, because we really locked down the maintenance fluid. So we said 1 mil per kilo per hour of maintenance fluid.
Right, that's what I wanted to ask you.
Preferably hypotonic, but if you don't like that, something different. So that was going to be low volumes in both groups. We wanted low amounts of unguided fluid, and then the remainder to be for the specific purpose of plasma volume expansion, obviously in the intervention group, guided by those stroke volume numbers. In the control group, guided by usual clinical assessment with whatever. So
1 mil per kilo per hour of 5% dextrose or dextrose saline.
Yeah.
But if you did choose to give Hartmann's, you could give that.
Yeah.
So 4 hour case, 100 kilogram person, that's 400 mils of maintenance fluid.
Yeah. Yeah.
But both groups got that.
Both groups.
You shouldn't give too much away about the fine print because the reviewers are going to want to comment on this. You've got a lot more work to do on it, but they're going to be really interesting things that we're going to be looking at to understand the results about timing of different things and volumes and doses and what happens next. Because if we may jump to the primary outcome, of which we ideally first like to know, you touched on this today, what was the power calculation predicting the primary event rate should be, and we should remind ourselves the primary event rate in a second. What did it turn out to be? And I think Desiree's given it away already, but
No.
wasn't there a difference?
If you didn't have to say that, no one knew that before.
Nobody would have noticed. It wasn't for those pesky
I'm going to
kids.
edit that out.
Okay, so primary outcome, post-operative infections within 30 days of randomization. Baseline event rate we took from...
It's a composite of all common infections.
So it could be triggered by the presence of one of a number of pre-specified infection types, which are all very standard definitions in line with Centers for Disease Control definitions, and they had to be of the Clavian Dindo grade 2 or above, i.e. you needed to have required an intervention for it, which of course you would for almost all infections. You'd start antibiotics, for example, and other treatments as required.
So, for example, pneumonia, surgical site infections, urinary tract infections, of which one can start to hypothesize about the links. But that's based on the prior meta-analysis which you presented, which showed that there was a signal there to suggest it was worthy of pursuit.
It looked like a good, important post-operative morbidity target that was modifiable. So we felt that was the best chance of seeing some improvement with this intervention. And the rate in OPTIMISE had been about 30% post-operatively. It was one of the secondary outcomes. And that was kind of backed up. That was consistent with a lot of the more observational series that were out there generally. So that was what we took to be the assumed baseline rate. And then the sort of the effect we thought we took again from the evidence syntheses, kind of a 0.8 hazard ratio. So that would take us down by 5% or so. So we thought, right, 30 down to 25%. It probably sounds modest to a lot of listeners, but actually, we're into the realms of marginal gains in perioperative care. We really need to find these important but modest improvements that you magnify them by the millions of people that have surgery, and actually you're seeing a big reduction in healthcare burden. So we felt actually, 5% reduction in post-operative infections. That's got a big patient impact. It's got a big healthcare impact. We felt, that's a good, feasible, and important difference to try and find. And that landed us at 80% power at 2,500 patients.
And you ended up with what event rate in the control group?
23%.
Okay,
almost exactly.
So we turn around when that happens and say you're at risk of potentially being underpowered or having some bias when you analyze the result.
And let me ask you that, Monty, because you didn't achieve the 30% of what it was.
Because your power calculation was based on the assumptions, but that power calculation, if there's a result there with a p-value and a big difference, it's almost definitely real.
Yeah.
If there's absolutely no difference, it's almost definitely real. If, as happened before with the previous study, it's kind of in the grey zone, you think, oh my God, here comes OPTIMISE 3.
But as if you're happy for me to
let's do
share
it
right now.
Yeah, before Monty has to puck up. Go.
The rate in the intervention group was pretty much exactly 23%.
So absolutely no difference at all.
Just not a whisper or hint of a difference in the incidence of post-operative infection. So you would, as you said, if you were seeing a small difference, you might be under.
No, I'm the crying.
I'm sorry. I'm sorry. It is what it is.
I know.
So I mean, we're happy with the power. I mean, there just is really no sign.
They push back.
The hazard ratio is just firmly across 1.0. And the p-value is like 0.7. So,
and normally then you look again. We've got to be careful with too much away. But you look around at everything else and you think, oh, I've got to sniff out some difference here that's going to give us some reason to say that we looked in the wrong place for the wrong thing. But apart from a hint of a signal of harm, which I think you're going to tell us about in a second, I didn't see, just glancing at it, I didn't see anything else where you'd say, well, hang on a second.
Yeah, no, that's right. So the component parts of the primary outcome, all those different types of infections looked pretty well matched across the piece in the two groups. Other morbidities, they weren't formally analysed, but we collected and report them. Other post-operative morbidities and the process outcomes, long-term mortality, that sort of thing, all seemed pretty much the same in both groups.
Monty, do you need to fly away?
I'll hang around for a little bit.
Okay.
Just because I want to wrap up this next little bit.
I know. Yes, go for it.
So you mentioned a safety signal.
Yeah.
What we did find, so we because there was a slight question mark about cardiac events after OPTIMISE. So a small number in the intervention group, none in the control group. Troponin's the same as per Mike Gillies' work, but that was a question we needed to answer. So we built in a safety outcomes of 24-hour and 30-day cardiac events, and that could be comprised of arrhythmias, MIs, myocardial injury, and so on. And we saw an increase in the intervention group. The overall numbers were not high, I'm going to be pushing myself to remember the exact figures that aren't in front of me, but it was something like 3% in the intervention group, 1.5% in the, like 38 acute cardiac events in the intervention group, 21 in the control group. And when we looked at the breakdown of that, it was really just due to an excess of early arrhythmias. And that kind of, that chimes with hindsight. You might think, yeah, maybe we were going to see some of that. It also chimes with the fact that about 20% of intervention patients required a per protocol reduction in the dobutamine infusion rate or stoppage of the dobutamine during the intervention period.
A few things to tidy up, if we may, Desiree, before we reach a conclusion, because there's already been stuff flying around on social media, as one would expect it to.
Has there?
In hours. It's only been
There's nothing in my Twitter feed. I don't know what's going on. Tell me more.
Social media comes in very many different forms.
I know, Monty.
The dark web.
Okay, so conflict of interest from me. I've been working with Edwards Life Sciences for a long time, but consulting for them for a year, and I think everyone knows now I'm going to be joining them soon.
I don't know if everybody knows. We had never officially announced that on Top Med Talk. So
joining them soon, stepping down from various different roles and responsibilities. Very proud to be becoming their Senior Vice President of Medical Affairs and Professional Education. And
congratulations.
Yeah, thank you. The Edwards relationship, and you happen to be called Mark Edwards.
Doesn't help, does it? It doesn't help. I'm not from Salt Lake City.
You might get to.
I'm from South London.
But you did receive one honorarium once for coming to a meeting.
That is right, yeah. 6 years ago.
The relationship with Edwards, as I understand it, was, because before my direct involvement time, was twofold. Was to do the trial that you and Rupert and others had designed based on OPTIMISE I, they went to an industry partner to see if they could get the technology required to consistently use that in the patients, which would cost a lot of additional money if you didn't routinely have it. So that was one, as I think, his contribution in kind.
Yeah.
And you were successful in securing an unrestricted grant. In other words, you applied for a grant process through the Edwards grant-giving body, and they awarded you a grant to match the funding that you were getting via an NIHR professorship. So it's yours trial with support from Edwards in that way.
Right.
Edwards the company, Life Sciences, not Edwards the family.
Not Edwards the person. Correct. Yeah. So I mean, it must be said that Rupert, as chief investigator, was the primary contact with Edwards and with those negotiations. So the fine detail I'm sure he's happy to share with. But yes, essentially, we proposed a trial design. We showed it to them. They were allowed to comment, but that we were in control of what we did. And as Rupert said earlier today, I mean, the relationship was very professional. There was absolutely no attempt to kind of interfere or to meddle with what we did. They were very supportive. They kept supporting through COVID. And again, this is a standard process that you do when you're working with industry. You establish the rules of engagement. You say, yes, we may show you the results to give you a little bit of time to prepare some responses before they come out, but you're not dictating what analyses we do. You're not dictating which journal it goes to or not. The investigators have the primary control of how the trial runs and, what the outputs are. And that's a really important point. And I think we have to be a bit mature about kind of working with industry and acknowledging that sometimes we should and probably, need to work with industry. And, absolutely, we should be wary of relationships that are unclear. We should be wary of, some of the bad old days where things were not pre-specified well, where there were fishing expeditions to go and look for results that weren't there, where negative trials just sat quietly and never got published. Everything has been super transparent and super robust in that way with this trial, as you would expect for any quality large clinical trial.
And the two other things sort of flying around already on social media you might anticipate is the one, in my opinion, relatively unsophisticated sort of umbrella summary that goal-directed therapy doesn't work, as opposed to what I think was drawn out in the POCI meeting is goal-directed therapy is an umbrella term for very many different approaches to choose various different physiological targets and interventions that may or may not make a difference to outcome. So my biased conclusion, which you may or may not agree with, is the stroke volume optimization followed by a fixed dose of dobutamine version of it being summarized as goal-directed therapy, we should not do based on the current available evidence. And mixed in with that is we're not sure anyone was doing it.
The latter, I don't know. The former, I would also say we should not do as a routine.
Yes, as a routine.
In the surgical population that we looked at.
Yeah.
And I think that's a really important point. We've got to recognize what a large, pragmatic clinical effectiveness trial can and can't tell you, and what it's trying to tell you. We're trying to inform widespread policy here, and what we should, do as our standard approach, as our routine standard care to large groups of patients. So if this has been a positive result, I think we would have been in a strong position to say to policymakers and to trusts and whoever else, we should be doing this as a fairly routine thing for most of these patient groups. And as the result is negative, we would be saying, well, actually, no, we shouldn't be needing to do this across the board as routine in all this patient group.
Every single patient.
That does not speak to, it cannot speak to whether those, that the biological rationale for this working or not is in some way flawed. I mean, that's just not what this can ever show. Do I now think that giving fluid boluses to see if there's an increase in stroke volume is not a good way of assessing fluid response? No, of course not. Of course, it's still, that paradigm is still, a perfectly valid one. Why would it not be?
So to close out my questions, immediate ones related to this, what I took away from the Perioperative Quality Initiative meeting results, I don't want to undermine those either, which had the goal-directed component of it, which you led, Mark, and the blood pressure conclusions, and to a certain extent the fluid conclusions, were that if you think a patient is hemodynamically unstable, trying to determine the main contributory factors, whether they be preload, i.e. volume requirements, or afterload, i.e. a vasopressor, or contractility, i.e. an inotrope, trying to determine whether that's the driving cause and treating the underlying cause or causes is still a solid basic tenet.
Right. And I think we need to, it's quite useful to look across to critical care and think about how the story's evolved in the early sepsis resuscitation, goal-directed resuscitation story that showed early promise, and then promise and arise, and others showed actually, again, using this routinely in unselected patients with sepsis may not be any better than routine care. Fine. So let's not do it for everybody as a routine. Do my intensive care colleagues still have cardiac output monitoring on their units? Yes, they do. Do they still reach for it if they're unsure of a patient's hemodynamic status, that they're getting into difficulties with where we are with fluid balance and hemodynamics? Yes, they do. Of course they do. So who knows? But, that it's interesting to look at that parallel and see how that might now play out potentially.
So I'll let you take back over, Desiree, because I think that we talked to you recently about your practices that you've been doing for a long time under the label of goal-directed therapy. Yeah. And I think I've reviewed your algorithms with you fairly often recently. They don't look anything like the OPTIMISE II protocol.
No, they don't. And then, we had a lot of discussion about that during POCI, which we'll flesh out in another conversation. But, we're not using dobutamine. We look at, different parameters as a collective and reach for targets on those. And it's just a very different approach. Our goal-directed therapy approach is very different.
Which may or may not work.
It may or may not work.
Seems to work for you.
It does. And honestly, that we derived our algorithm based upon what we kind of were already doing for our what was in our enhanced recovery pathway. And it just evolved as we had conversations about what were we already doing. So we didn't want to make it too different than what we were already doing. And as we have access to technology for more patients to be able to utilize and see if this works, adding in other, some other parameters that we hadn't necessarily looked at before. And, it's working. And everybody's fairly happy with what the algorithm looks like now. So we're testing it out.
So we know it's inevitable. We're always going to get another negative trial, which very many of us get upset about these things with the huge amount of work that goes into them. Because it's answered a question. Our good friend John Myberg, when he's pressed on releasing the results after, before, he's poker faced and says, one thing I can tell you is we will answer the question. So you've answered the question.
Yeah.
The important thing for people to do is to focus on what was the question.
Yeah.
And we look forward when the publication comes out
to
seeing all those extra nuances that we get a chance to discuss. So I'd like to congratulate you and the core team.
Yeah.
I was very much on the peripheries of it, but very core team for getting an amazing study over the line. Something to be very, very proud of.
Yeah. No, I mean,
it's another thing we know not to do.
Right. Yeah. And just on that, I think, just to take the opportunity again to acknowledge the huge effort internationally that's gone into this.
Yes,
and,
it was incredible.
For me, the mentoring from Rupert has been amazing. It's been a great first big trial to work on. But the international effort has just been phenomenal to keep it going through COVID without even stopping.
That's what I was going to say.
Is just amazing. So there's a lot of people that own this, and they should be rightly proud of the contribution they've made.
Yeah.
Floella,
yeah. So nearly done. So, if you're interested in a non-inotrope involving GDHT algorithm, okay, it's different. It's emergency abdominal surgery.
Yes.
It is mostly going to be open surgery, although, lap stuff's in there. It's because it's linked to our national laparotomy audit. Yeah, we're nearly there, but we need some help. I mean, we've recruited 2,850-ish of a target of 3,100.
Oh gosh!
So we are so close. But the teams are struggling out there. It's hard times out there. So we're nearly there. But that will, I think, the fact that's a fluid only, again, very similar algorithm to OPTIMISE II, but no routine inotrope, and again, similar broad hemodynamic targets around the map. It's going to be really interesting data. We're nearly there, but we just need a bit of help getting across the line.
Yeah, no one's having a laparotomy surgery these days. But...
There's loads. It's just hard.
It's hard.
Emergency surgery recruitment is.
I can imagine.
But it's again some amazing effort out there from the teams. It's it's hard work.
Adaptive platforms. Let's leave it there.
Yeah, I mean that's it's tempting, isn't it?
The next
complex,
the next frontier.
Complex, not cheap. Not without complexity. It's a
not
topic of interest.
Not that major RCTs aren't either.
Right. Yeah. I mean,
time out on that one.
Time out on that one. But it'd be great if it didn't take 10 years to take each step forward in the evidence base.
Yeah. Well, congratulations. And I have to commend you, Mark. Your level of professionalism and that poker face.
The pokey face. I put my pokey face on. You missed a trick there, Desiree.
That's funny. So a testament to the fact that it's been a long 3 days. My brain is a bit fried at this point. But through the entire time, we had, our whole session was around goal-directed therapy. And I mean, it was, I'm sure, on the tip of your tongue, but we had a lot of it wasn't heated at all, but a lot of really intense discussion. And never one time did I ever feel that you had, let the cat out of the bag or anything like that.
I'm kind of simultaneously proud and a little bit scared of myself now in how able I was to do that. Maybe everyone's a little bit disturbed by it now.
Crime.
But it's worth just explaining the rationale there. And it might, to an outsider, seem a bit nuts that we knew this information and we were going to spend 2 days establishing some consensus statements. Why
when it came out the next day,
when it was just about to come out, it was really important. And we had a strong view that actually we really, really wanted to respect the work of the OPTIMISE II investigators around the world, and we didn't want it leaking out. I'm sure people would have been really good in the room, but actually we just didn't want it to leak out. And then somebody hears on social media or something, oh, I heard it was in that. If you're sat there in wherever. And you've worked some people worked really hard, and somebody's found out the results before you, and they weren't even involved in the trial. That sucks a little bit. So cool. we were very keen and we've worked out a nice process with the group whereby we've looked at some potentials of how we might play things depending on the results. We're going to have post meeting meetups.
We actually did come up with a consensus statement on goal-directed therapy for major elective abdominal surgery.
We parked it. We put a placeholder in there, and there's going to be a bit of debate now, but I think that's fine, and I think it will play into the POCI recommendations as I think it needs to.
Yeah, for sure.
And there's a process for that, so it's been a bit different. But I think it'll work out well, and as I say, that's kind of the rationale.
Yeah, and I have to say that at first I was seriously bummed about it.
Yes.
Let's talk about what I was bummed about. The first thing I was bummed about is the fact that we didn't have it when we're sitting there having these conversations. Right. I think we walked away with actually better for it.
Yeah.
Yeah, it's an interesting.
I wasn't in the room, but the results better, I think.
Yeah, it was
you
an
can
interesting thought experiment.
Yeah, you haven't been distracted
Right.
Yeah.
by
Yeah.
the chaff in the water.
Yeah, exactly.
Yeah.
Because it could have, I guess, influenced all the other stuff that I think has been really good that we've been able to say about GDHT.
Yeah. So too.
So I think it's worked out well.
Yeah.
But I would say that,
good.
wouldn't I?
Yeah, of course, the chair. But it really was. It was good exercise. So, Mark, thank you so much for taking the time to do this. Thank you to you, all of your colleagues, for the hard work that we know that this was. I think it, will answer some questions and opens up a lot more. And, so here we are.
We'll see.
Keeps on rolling.
Submission is ahead. Obviously, it's coming through peer review.
Yeah.
There'll be some ironing out of the little details in the data, so on, and let's see what comes out of the peer review process, which we know is an important part of this as well. So
you got the 6-month mortality follow-up, have you?
That's in there. Yep, that's in there.
In already? You got it? Okay.
180 days is in there.
Okay.
Yeah, no difference.
Oh, wow.
No difference.
Okay. That's cool. All right. Well, we'll follow up back on it. I'm sure we'll have some more conversation around it whenever we have the pokey discussion. Mark, again, thank you so much. And thank you for listening to Top Men Talk. You can always find us at topmentalk.com on your favorite podcatcher and social media platform as well. We are there. Check out these conversations. Please do go back and listen to Rupert when we talked to him at the ESA.
Before he had a clue about the results.
He didn't know. He obviously.
We've had these for like 3 weeks. It's really fresh.
Yeah. And we great conversation with him. And over the years, we've had others about this hot topic. So check it out. Thanks so much for listening, everybody. Cheers.
Bye.
Top Med Talk. Thanks for downloading Top Med Talk. Don't forget to subscribe via your podcatcher. Don't forget to check us out on social media. We're on Twitter, Facebook, LinkedIn, and YouTube. And also, don't forget, Top Med Talk is the broadcasting arm of EBPOM, evidence-based perioperative medicine. We'd love you to find out more about that. If you check out ebpom.org, you can find low prices on some of the conferences we're organising around the world. Many of them are virtual and don't even involve you leaving your own home. Check out ebpom.org now.